the bulk of my presentation focuses on the idea of lowering the concentration of these pathogenic proteins. And that's happening in HD and coming very soon to a neurogenetic disease near you. But the other half of the other side of the coin is this repeat expansion concept. And the long-held view that, in most of these diseases, we're dealing with a triplet repeat, and the genetic test is to count the number of triplets. And if it's above a certain size, then that's a positive test. And broadly speaking, bigger triplet sizes is associated with earlier onset and faster progression. So, and that's all fine. But it's long been assumed that that was a static thing. So you have your genetic test at the age of 25, say, your CAG repeat, if you've got Huntington's disease, might be 42. And that 42 is that's your life story, right? That will determine your age of onset to whatever extent your age of onset can be determined. It's a convenient way of thinking about it, but the reality is that we've known for years that in the brain, in certain cells of the brain and other tissues, that CAG repeat can actually expand during life. So some cells in your brain might have CAG repeats of 70, 80, 150, and this is called somatic instability. And we knew it was a thing, but we didn't know how important it was until we did big GWAS studies in Huntington's disease, and then looked at the results across the triplet repeat diseases and basically found that, generically speaking, somatic instability is a huge thing, and single gene differences that contribute to somatic instability also contribute to earlier onset of the disease, and are also, broadly speaking, all linked with DNA repair. So in other words, your DNA gets damaged, or this is the idea, your DNA gets damaged, and the cell tries to repair it. In the process, if your DNA repair equipment is put together in a particular way, it can repair it erroneously in a way that adds extra triplets. And when that happens, bad things occur, and your disease is likely to happen earlier. Conversely, there are protective variants where the DNA repair becomes more reliable and repeat expansions are less likely to occur. But what we're left with is a situation now where these GWAS studies have revealed a number of potential targets outside of the genes that cause the disease directly. So in Huntington's disease, this is not targeting Huntington protein or the Huntington RNA, but rather targeting the DNA repair genes, the genes that make the DNA repair proteins. And it's at least theoretically possible that those treatment targets could prevent the onset or delay the onset of these neurodegenerative conditions, and could also act across multiple diseases because these are shared mechanisms of somatic expansion. So this is a relatively new field, but already there are companies and academic groups that are working on both small molecule and ASO approaches to modulate the process of DNA repair or the expression of certain DNA repair proteins. And doing that, particularly if applied early in these neurogenetic diseases, is, I would say, an approach that has great promise. Because if you think about the way that Huntington lowering or the lowering of toxic proteins works, you're essentially reducing the number of toxic molecules. But if you can prevent the triplets from expanding, what you're actually doing is pre-detoxifying each molecule of that protein. So you're not reducing the concentration of mutant Huntington, say, but you're making each molecule of Huntington less toxic than it would otherwise have been if the CAG had been allowed to expand. So that's the other the second bit of my talk focuses on this concept of targeting somatic expansion of disease-causing triplets.